This lesson's on carbohydrates and specifically polysaccharides. You'll be able to describe the structure and function of amylose, amylopectin, glycogen, and cellulose. Now, the first thing to recognize is that all four of these molecules are polymers of glucose. They're formed by joining different glucose molecules together with covalent bonds. But what makes them distinct from one another is specifically what type of glucose and how the glucoses are connected. Let's start with amylose and amylopectin. Both of these are what we think of as starch. They are energy storage, or their function is energy storage for plants. But they differ in just how they're structured. So just a reminder, when we talk about glucose, it's a 6-carbon ring, or rather there's 5 carbons and an oxygen in a ring and a 6th carbon sticking up. And we number the carbons like this: 1, 2, 3, 4, 5, 6. And alpha glucose has a hydroxyl group pointing down from the ring plane on carbon 1, and beta glucose has that hydroxyl group pointing up. Otherwise, they're identical. So that's a review of the difference between alpha and beta glucose. Also remember that when we join carbon 1 from one glucose to carbon 4 of the next one, we would call that a 1,4 glycosidic linkage. In contrast, if we joined carbon 1 to carbon 6, that would be a 1,6 glycosidic linkage. So with that reviewed, amylose is formed by 1,4 glycosidic linkage, 1,4 glycosidic linkage of alpha glucose, or multiple alpha glucoses. And that gives it an overall helical structure. So it looks like a corkscrew, like that. Amylopectin is also helical, also forms that kind of corkscrew, but it's branched. So it branches off like that in multiple places. So there's like corkscrews coming off of corkscrews. And the way that it does that is it has both 1,4 and 1,6 linkage, glycosidic linkage of alpha glucose. So that additional way of joining multiple glucoses provides that branching structure. And in fact, the branching gives more endpoints where enzymes could put on or take off glucose, and so enables glucose to be added or taken off of amylopectin more quickly. But both amylose and amylopectin form what we think of as starch, and in fact starch is a mix of the two, usually more amylopectin. Here's a molecular model of amylose showing its helical structure. Carbons are in gray, oxygen in red, and hydrogens are in white. And here we're looking at a model of a branching point in amylopectin. We see two helical structures branching off from each other. Glycogen is very similar in structure to amylopectin. It is these kinds of helices coming off of each other, and it is also 1,4 and 1,6 linkage of alpha glucose. The branch length is a little bit different. It's shorter with glycogen, and the function is here energy storage in animals. So you can really think of glycogen as being like the animal version of starch, and predominantly glycogen is stored in the liver and in the muscles of animals. And in humans, for example, we've got about a day's worth of glycogen before we would need to start using other fuel sources. But it's a energy or fuel storage in animals, again, similar to amylopectin. In contrast, cellulose, which is a structural molecule, it makes up the cell walls of plants. In fact, if you wear a cotton shirt, you're wearing cellulose. It is formed by 1,4 glycosidic linkage of beta glucose. And that small difference means that it's not helical, but it's actually flat. And so the 1,4 linkage forms these fibers like this. I'll show you those below. So this would be 1,4 linkage of beta glucose monomers, and in order to do that, every adjacent glucose is actually flipped upside down from its neighbor. So if you look at the highlighted OH here, or the CH2OH, you can see they're upside down in each neighbor. And that means that it doesn't twist in one direction. It sort of twists one way a little bit and then back, and that keeps the whole molecule flat. So you get these long fibers, and in fact, they can join with their neighbors by hydrogen bonding that I'm drawing in red here. And that forms these cables that are very, very strong. And that gives strength to the cell walls of plants. So here's a look at part of a microfibril of cellulose, and you can see when you get a look at it end on that it's just flat. Here we're looking at the effect of 1,4 glycosidic linkage of beta glucose. So that's an introduction to polysaccharides.